This is a recording of the Lab Practical 2 study guide for Bio 111, starting with Lab 11 on mitosis. The first thing it says is to be able to identify cells in all 5 stages of mitosis, which would be, and if you look in your lab manual on page 152, you can see some pictures. The only ones you'll have to identify on the lab practical will be the onion root tip cells. So I know we looked at some whitefish blastula cells, but it's the only ones you have to worry about are the onion cells. And you'll need to know, need to be able to identify cells in interphase, prophase, metaphase, anaphase, and telophase. So I've got a little slideshow for you to help you with that. And if you forget the stages, if for some reason you're on the lab practical and you forget them, well, number one, it's going to be multiple choice. So you shouldn't, you should have no problem remembering, but IPMAT will help you remember interphase, prophase, metaphase, anaphase, telophase. So this is a picture that I just found online of cells in different stages. The only one I don't see on this particular image is anaphase. So let's start out with interphase. Remember, and this is an I. This little red wavy letter is supposed to be an I. So you can tell I made this slide. But these two cells are in interphase, and if you ever wonder if cells are in interphase, look around on the rest of the, when you're looking through your microscope, look at the rest of the cells in the field of view. Most of them are in interphase, so the ones that you see the most of are the ones in interphase. But you're going to notice, like this one down below here, you're going to notice that the nucleus is round and it's definitely got a nuclear envelope surrounding it. You know, you see that because there's, it's just very smooth. But you also can see even darker structures on the inside of the nucleus. This is a nucleolus, and sometimes you'll see more than one nucleoli. But you can see that the nucleus is intact, and it is pretty much shaded in a solid dark color blue. You can't see individual spaghetti strands like you can here, for example, in this cell. So this is going to be when the DNA is in the form we call chromatin, and it's very spread out and diffuse. So it doesn't condense and form those structures we call chromosomes. So those are going to look like spaghetti strands under the microscope. So I'm just going to point out a few cells that are in interphase. You've got the ones that are labeled here, but then there's two kind of right beside. Those are in interphase. Up here, this one, and this one are all in interphase. So these are cells that are clearly in interphase. This one down here is in interphase, and you can see the nucleus is the larger blue structure, and the nucleolus is darker, and it's inside the nucleus. So that's what you're going to see for interphase. Prophase, you're going to start to see the integrity of the nucleus is going to start to be breaking up, because during prophase, the nuclear envelope disappears. It dissolves or breaks apart into the cytoplasm. And you're also going to start seeing chromosomes instead of chromatin. So you see the little spaghetti strands. So here's a cell in prophase. Here's another one in prophase. These are clearly in prophase. So that's what you're going to look for a cell to identify it as prophase as compared to interphase. And then we have metaphase, and all the ones with a yellow star are in metaphase. I know sometimes because what we learn about metaphase is the chromosomes are lined up at the equator or the middle of the cell. Well, they're spaghetti strands, so they're not going to form just a straight band. They're going to, all of those chromosomes lined up at the middle are going to kind of look like that. They're obviously together in the middle of the cell, but because they're spaghetti strands, they still look kind of jumbled. But this one is in metaphase. This one is obviously in metaphase. This one down here is still kind of disorganized, but you can tell those chromosomes are lining up in the middle versus completely scattered in prophase. So you've got 3 that are in metaphase. Each one that has a gold star is in metaphase. I don't see an anaphase, but I do see a telophase, and I've labeled that one here. I circled the group of basically it's one cell, but it's about to divide into 2. So what you're going to look for in telophase is the chromosomes have stopped moving. They've kind of balled up at each end of the cell and just formed a little ball. You can see the little spaghetti strands, but you can also see that the nucleus is starting to come back together. And then what you really look for in telophase is a cell plate, the formation of a very faint line or cell plate that will eventually be the new cell wall between the 2 cells. So this one is a darker cell plate. Sometimes it'll be lighter than this picture. But this one is in telophase. It's about to split in 2. It's about to undergo, it's actually in the process of finishing up telophase and cytokinesis also, which is when the cell splits in half. Okay, now we've got some here that are labeled that I found online. So starting with interphase. Now these are lighter, but you can see the ball shape, the spherical shape, and the nucleolus is very clear inside. If you compare that to prophase, you can see those spaghetti strands, those chromosomes that have condensed. Metaphase is really clear. 1, 2, 3 cells right here together that are in metaphase. There's a lot, actually. Here's another one. Metaphase, 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 metaphase. Then we have an anaphase over here to the left. You see how the chromosomes are now pulling apart, and they've just started pulling apart and begun to move toward opposite ends of the cell. So this one is in anaphase. This cell right here is in anaphase. But the one that's labeled over here is further along in anaphase. But you can still see that the chromosomes appear to be moving. It's like someone has taken a snapshot while those chromosomes are moving. So they look as if they're moving toward opposite ends of the cell. When you get to a cell that's in telophase, the chromosomes don't look like they're moving anymore. They look like they have stopped moving, and always look for that faint cell plate that begins to form in between them. So this entire cell, this whole entire cell here is in telophase. Here's one set of chromosomes, and here's another. You can tell they don't appear to be moving anymore. They appear to have stopped, kind of balled up together. And then you see that faint, very, very light cell plate. All right, on this page, I'm just going to point out some that I see that are clearly in certain stages. Interphase is very obvious here. This interphase, 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 interphase. All these that have these solid nuclei, this one is interphase, interphase. All of these here are interphase. And then for prophase, this one is in prophase. This one over here is clearly in prophase. This one clearly in prophase. The nuclear envelope has completely disappeared, and you can see those scattered chromosomes. Metaphase, that's a metaphase. This one is an early anaphase. You can see that the chromosomes are separating here. This one is anaphase. This one is anaphase. All right, now this cell is clearly telophase. This entire structure here is one cell, but it has started dividing. And you see the 2 sets of chromosomes that have stopped moving. and you can see the formation of a cell plate in the middle. I'm just going to look real quick and see if I see another telophase maybe. Probably, but that's a good one right there. So, and here is, I think, the last slide that I posted. Here's a good prophase right here. This is one that's definitely in prophase. Interphase, interphase. There's a prophase. This is a prophase. Prophase, prophase, prophase. Let's see. Let's find metaphase if we can. Here's a metaphase. This one appears to be metaphase. Let's see. This one is anaphase. This cell right here is anaphase. And then I see two cells to the left of this one that are in telophase. This one is telophase. See the faint cell plate there? And then this one, this cell, is telophase. You can clearly see that cell plate formation. Oh, here's some more. Okay, anaphase. This cell is anaphase. Interphase, 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 metaphase, prophase. This one is at like right at the end of telophase. It's almost completely divided into, that cell plate is really almost a complete cell wall at that point. But this one is definitely anaphase here. Okay, all right, and now to say that. Okay, number 4 on page 155. Let me find where I got. Okay, that is this question right here. Now my pages are not numbered, but this is the question that it's talking about. It's in your lab manual, and hold on, I was going to blow it up some. All right, it says identify the following structures as animal mitosis and or plant mitosis. So you're going to say AM for animal, PM for plant, and then you could do both. All right, so centrioles are only going to be seen in animal cells, so AM. Cleavage furrow is only going to be seen in animal mitosis, where the two cells start to pinch in two instead of a cell plate forming. And plant mitosis will be a cell plate, and then metaphase, anaphase, telophase, interphase, and even prophase are going to be both, because all of those stages happen in both animal and plant cells. Okay, back to the study guide. Let's see. All right, lab 13 on heredity. It's giving you some Punnett square problems. If you will just be able to solve for incomplete dominance and codominance, if you'll just be able to solve the ones from the jack-o'-lantern genetics, but the ones for sex-linked and or X-linked and blood